ठीक है। Welcome ji, my name is Platavesh. We are for the market talk on supercomputing and machine learning for big biomedical data by Usman Tariq. He is alumni of UAT and he completed his graduation in electrical engineering in 2014. After he completed his master's in computer engineering from Western Michigan University. Currently, he is pursuing his PhD in computer science from Florida International University. His research interests includes deep learning, parallel programming, and high-performance computing. I think I will hand it over to sir. Please.
Thanks, Bilal. Hi, everyone. My name is Usman Tariq, and I'm a third-year PhD student working with Professor Fahad Saeed at Florida International University at the Department of Computer Science. I'm going to start with thanking Bilal and everybody else who helped coordinate this event. So let's just begin. So the talk is going to be about supercomputing and machine learning for big biomedical data. So we are essentially high performance computing and machine learning lab, and our domain focus is computational biology in different subfields within bioinformatics. So just the overview of our lab, what we do. So we are NIH and NSF, which is National Institute of Health and National Science Foundation funded lab. The lab advisor is Professor Fahad Saeed. We are working on designing parallel computing machine learning algorithms for big scientific data, which is mainly focused on bioinformatics and biomedical data. And our focus is using machine learning to improve the algorithms or improve the performance and scaling the algorithms for large supercomputers. So we are actively working on, let me get this pointer. We are actively working using supercomputers. So this one is 𝐒𝐮𝐩𝐞𝐫𝐜𝐸 supercomputer. This is a previous version, Stampede. Then we started working on Comet, and now they have replaced it with 𝐶PANs. These are just the latest version of the supercomputer with like thousands of cores and GPUs. We have servers with the latest GPUs and FPGAs in our lab. So this is kind of like the infrastructure of our lab. So this is the domain, big scientific data, and we are working on machine learning, deep learning. That's my project, mainly for proteomics data, which I'm going to talk about in a minute. And then we are working on big data analytic techniques. My colleagues are working on that, and then parallel algorithm, which are kind of mix of both. Deep learning uses GPUs, and we're also working on distributed computing. So that's kind of like the overview of our lab. So the main focus of our research is to design scalable algorithms. So scalability, you can think of as your algorithm should scale with the increasing data set size and also with the available number of compute units, the CPU cores, or having multiple GPUs or any other device. Machine learning is relatively new. So we have been working on machine learning for connectomics. Connectomics includes fMRI data, brain scans, and other stuff. So we have been working on that using machine learning for a while, but for proteomics, we recently started working or designing machine learning and deep learning algorithms. And then our eventual goal is to enable precision medicine. That's, you can think of it as the punchline or the eventual goal of our lab, the kind of like the motivation that we want to enable precision medicine. So we have to take care of a lot of things, designing scalable algorithms, dealing with big data. So bioinformatics, proteomics, genomics, connectomics, and other omics data is essentially considered big data nowadays. We have terabytes of raw data available that needs to be processed efficiently, and we want to enable, ideally, we want to enable real-time processing. So again, machine learning, deep learning, high-performance computing, and then incorporating the domain knowledge, which is these mainly these three things. So moving on, these are the three main goals of our lab. So as I mentioned, the precision medicine is kind of like the generic overview, but specifically what we are focusing on is that we want to, so for example, in proteomics, you sequence proteins, sample specific proteins, you take a protein sample, you do mass spectrometry, you get spectra, and then you process to sequence a protein. And one of the big applications of that process is to understand cancer and HIV better. Because even though cancer is usually related to gene mutations, but eventually what they do is they produce newer proteins which we haven't sequenced before. So being able to sequence those proteins and understanding their functionality is crucial, and that's where proteomics field is where we are working on. This has been our past project and probably will be the future project, identifying unknown and non-bacterial and viral pathogens, which again translates to genome sequencing, protein sequencing, and then eventually designing algorithms for those data sets. So our focus is not mainly on the biology part of it. We normally train students to have the basic knowledge or as much knowledge as they need, right? The literature review, but eventually the focus is designing better algorithms. So, and then we are working on early identification of mental disease. So this is pretty interesting project. So what we do is we take fMRI scans, the brain scans, and we do image processing or image classification using deep learning. And our goal is to identify ASD and ADHD and PTSD using these brain scans. So what is happening right now is that these mental diseases are identified qualitatively, right? A person asks you a bunch of questions, the doctor, obviously, and they tell you, okay, you have ADHD or you have PTSD, and that is pretty biased, right? So we want to have a mechanism so that we can quantitatively identify, like, what's the probability that a person has ASD, ADHD, or PTSD, and using fMRI scans is a pretty good start. And we've developed algorithms that are so far state of the art in accurately identifying these problems in children and adults. So this is a really interesting project that we are working on right now. So I'll give you the overview of 3 main projects that are going on in our lab. So one that I just mentioned is identifying ASD, autism spectrum disorder, and ADHD from fMRI images. So this is what we have so far, 80% accuracy. So it's classifying whether a person has ASD or not. And this was recently cited as the most accurate algorithm so far. Machine learning and high performance computing algorithms for mass spectrometry based proteomics data. This is the project that I am working on. So this project right now is subdivided into 2 projects. So one that focuses on machine learning and deep learning for sequencing proteins using mass spectrometry data, and the other one is designing scalable algorithms using high performance computing. So we are working on the MPI, message passing interface, so that we can run these algorithms on supercomputers. One of my colleagues is working on that part of this project. So our eventual goal is going to be that we can kind of combine these 2 projects into having a distributed machine learning algorithm for sequencing proteins using mass spectrometry data. So I'm going to talk about this project, especially the machine learning part of it, since this is my project, in a little bit more detail later on. And then we have this 3rd project. I worked on this project during my master's, but then during my PhD, I switched to this project because I personally got bored, but this is a really interesting project. So the idea is that we want to use reconfigurable architectures like FPGAs. So you guys are aware of FPGAs, right? The field programmable gate arrays. So what happens is that you can reconfigure the architecture or the hardware, right, for FPGAs using hardware design languages, Verilog or VHDL, or there are more advanced languages available. This can give you a lot of advantages, especially for machine learning, that they can speed up the process. And also with machine learning and deep learning nowadays, there is a big problem of energy consumption, right? So scientists have been training huge models with billions of parameters, but they usually tend to ignore the energy cost related to those model trainings, right? Because GPUs consume a lot of energy, and deep learning algorithms or models are trained on GPUs. So this kind of like a step in the right direction, at least for reducing the energy consumption and having more energy efficient architectures. So I'll go through these 3 projects. I'll briefly talk about this one, then I'll go a little bit more in depth for the machine learning project, and then I'll touch on the reconfigurable project. So, okay. So brain disorders are prevalent, especially in the developed countries and also the developing countries. So this image is kind of like the rolled out version of an fMRI. So you have multiple brain scans at different levels. But the way we treat this data is using 3D pixels. It's called voxel, right? So it's kind of like image classification, but you can think of it as 3D images. And there have been a lot of work in processing these for image classification. For example, Google recently designed ImageNet, and there were others. FaceNet for face recognition. So you can essentially use those algorithms or kind of like retrain them for identifying or processing these brain scans to classify brain disorders. So as I mentioned, that this is not my area of research. This is there to kind of like give you the exposure of the project that we have right now. Um, so yeah, one of my colleagues she graduated last year, so she developed this algorithm. Right. So one of the things that she was working on is that she designed a deep learning framework for autism detection. Uh, this is visible, right? It's pretty dim here. Anyway, so one of the techniques she developed is that data augmentation. We had a limited amount of data for training the deep learning algorithm. So what we came up with, that we did linear interpolation to augment the training data so we can have improved accuracy of the model. So one of the things that we designed is that kind of removing the noise using the autoencoders. I think we also used concrete autoencoders. I'm not sure about that one. And then we designed different deep learning architectures that I'm not going to go into details about that. But we did end up having pretty good performance. So this is the one, ASD Diagnet. You can look up, the paper is published and the code is available. And it ended up outperforming most of the previous state-of-the-art algorithms. So even with no data augmentation, you can see the performance is pretty lower than with data augmentation. So designing the data augmentation technique and then designing the deep learning architecture gave us that advantage. So that's pretty much it. If you have questions regarding this project, I can answer as much as I know. I can put you in contact. If you're interested, I can put you in contact with the person who is working on this project. But the next that I'm going to talk about is the mass spectrometry based proteomics. And using deep learning to sequence proteins. So that's my project that I'm working on. I've been working on for the past 1.5 years. So I'll give you a little bit of background of what mass spectrometry is and how we use mass spectrometry data to sequence peptides or proteins. So what happens is, say you have this protein mixture and you use enzymes, for example, trypsin. So the process is called tryptic digestion, to break the protein into smaller peptides. So it breaks. So okay, before I go on, let me kind of give you the overview of what proteins are. So proteins are made up of amino acids. Similarly, the way genes or DNA is composed of 4 nucleic acids, right? A, C, G, T, right? We studied that in, I don't know, physics or FSA. So proteins are made up of amino acids, and there are 20 of them normally. And we represent each amino acid using an alphabet. So M, E, S, and each of these letters represents an amino acid. And this entire string of these amino acids makes up a protein. So, but we don't know these proteins. I've kind of sequenced this protein just to kind of visualize it, but we have this biological mixture, right? A protein mixture, a solution. It can be a blood sample, a muscle sample, or any tissue sample, right? And we want to know which proteins are in there. For example, you have a cancer tumor, right? And there are proteins that we don't know their functionality, or they're novel proteins that are causing the problem, right? So we want to identify those proteins. And one of the most common method is mass spectrometry. So let's follow the process. So you take the protein mixture, this is a cylinder, right? A cylinder. And you do tryptic digestion. You put trypsin enzyme in it, and it breaks the protein into different peptides. Then you take the peptide mixture and you do mass spectrometry. So what happens in mass spectrometry is that mass spectrometer selects peptides, like filters them out based on their mass, right? So each peptide has different mass. They can be close to each other, and the mass spectrometer has a certain uh, accuracy threshold, but it selects the peptides within 1 Dalton window. And then what happens is that you break down that peptide. So you have multiple copies of the same peptide, right? And you filtered it, then you break it down at each peptide bond, right? So for example, you have this peptide and you break it at bond between S and G. So you will get 2 fragment ions. So one where there is only one amino acid S, and the other one where you have the remaining amino acids. And you call this fragment ion B1, and you call this fragment ion Y9. That's just a terminology that they use. And subsequently, another copy of the same peptide might break at this peptide bond, then this, and you have all the possible fragment ions of the same peptide. That makes sense, right? Now, what happens is the mass spectrometer detects these fragment ions based on their mass to charge ratio. To simplify things, let's just assume the charge is always 1. So we can say the mass spectrometer detects these fragment ions based on their mass. And here on x-axis, you can see that this is mass. So it's m over c, mass to charge ratio, but we're just going to consider it for now just mass. And you get different peaks corresponding to different B and Y ions based on their mass. On y-axis, you have relative abundance, and this varies based on the detector in the mass spectrometer or the chemical properties of the fragment ion. So the height of the peak is not as important as whether a peak exists or not. So to simplify things, we can just convert this into ones and zeros. So for each m over z value, for each mass value, you can consider whether a fragment ion exists at that mass or not. So this is what we get out of a mass spectrometer, a list of floating point numbers, right? So this mass is represented in floating point numbers, and then you have another column of their relative abundance. Now that's what you get, and you get millions and millions of these spectra. And our goal is to take a spectrum and identify which peptide it came from, right? Because mass spectrometer took the chemical peptide and broke it down into the spectrum, and now we want to process this spectrum to identify the peptides. That's our goal. That's where we will design the sequencing algorithm, which will kind of take this as an input and give the peptide as the output. So this is the base knowledge for being able to design algorithms for proteomics. So how do we actually process this spectrum to identify the peptides? So the idea is that you take this spectrum. So this is the same spectrum as you saw in this previous slide. It's just different peptide and have different peak intensities. And you take the peptide and you have different search engines. So this is one of the most popular methods to identify proteins from spectra. So what happens is we already have a lot of information regarding the proteins that we've already identified. For example, we have a pretty good coverage of human proteome, right? So we don't want to kind of process each and every spectrum and try to identify the protein that it came from, because it's highly likely that we've already identified that protein before, right? So what we want to do is we want to search that spectrum in a protein database, right? And there are websites available that, for example, UniProt, that can give you all the proteins that we have identified for different species. So what we do is we take that protein database and we use one of the search engines. These are the algorithms that the traditional computational algorithms that process this spectrum and search that spectrum in a protein database and give you different versions of peptides with different confidence score. And based on that, then you do post-processing to identify which of these peptides is likely to be accurate. So one thing I want to stress is that this is actually a real spectrum that you get from a mass spectrometer, and you can see a lot of peaks, right? Only less than 10% of these peaks are useful. So you have to do a lot of pre-processing to remove the noise, and even if you remove the noise, you cannot realistically remove every noisy peak, right? It's a stochastic process. So this entire algorithm is not going to be a deterministic algorithm. There is always going to be a probability associated with every output that you give. So this method, let's see how we actually do protein database search. So this is a reference proteome. You can think of it as all the proteins that exist in a human being, all the proteins that we know so far. And then what we do is we do in silico digestion. In silico is a term for non-computer science people, so which basically means inside the silicon. You to have an algorithm to break this protein into different peptides. So now this is happening in a computer. So you have a text file with these characters and you generate these different peptides. And then you simulate the mass spectrometry process, right? The same way we did here. But this was inside a mass spectrometer, right? This was a physical process. This is a computer simulation. So you take the database, you generate these simulated spectra, and then you take the actual peptide that came from the mass spectrometer and you compare it against all, well, a subset of the simulated spectra. And the comparison process can be different methods. You can compare peak by peak, whether a peak at a given mass value exists at the same mass value in the theoretical spectrum, or you can just do a dot product or a cross correlation to get a score of the matching accuracy. And that gives you different matches, and you pick, ideally you will pick the highest scoring theoretical spectrum that match with the experimental spectrum. And then because you know that this theoretical spectrum came from a certain peptide, because we did this in a computer simulation, right? You will say that it's highly likely that this experimental spectrum also came from the same peptide. So that's how the traditional algorithms work. So the problems in these algorithms are the simulation of these theoretical spectra is oversimplistic and can miss important pieces. As I mentioned, that when we do pre-processing of these experimental spectra, we can miss out on a lot of peaks that we thought were noise, but were actually really useful. On the other hand, we can actually miss some of the useful peaks. So this is one problem or challenge that the traditional algorithms face. And the second one is that this comparison happens is there are different methods using a dot product or cross correlation or other methods that exist that compare this experimental spectrum with the theoretical spectrum, right? And based on which score you use, you get different accuracy, and you can get different peptides associated with the same experimental spectrum. So one algorithm can tell you, okay, so this spectrum came from this peptide, which is the correct one. Another algorithm can say, oh, it came from another peptide because it's scoring that peptide higher than the original one. So it's the accuracy or the correct output is highly dependent on the comparison algorithm that you use. So what we decided is that we have enough data at this point where a correct peptide has been identified for a given experimental spectrum, right? And there are online repositories which have terabytes of labeled data, right? So why not we design a machine learning or deep learning architecture which can compare or learn a similarity function, right? So this comparison measures the similarity between an experimental and theoretical spectrum. So why don't we learn a similarity function using a deep learning architecture, which is highly likely to be more accurate than the handwritten ones. So I'll show you the network architecture that we came up with. So as expected, it has two branches, right? One branch is going to process the spectra that came from mass spectrometry, right? It'll take the experimental spectra. And the other branch will take the peptide strings. So as you might notice that in the previous method, we took the peptides, right, from the database and simulated theoretical spectra. So right away, we are eliminating the simulation step, right? We are taking the peptides as it is, and we want to kind of do some process here, which I'll explain, so that we can directly compare the experimental spectra with the peptides. So what happens is you take the experimental spectra, so this is just a fully connected layers. Um, and what you do is you kind of embed this spectrum based on their M over Z values into big arrays, right? Big floating point arrays with values that are normalized from 0 to 1. And then you process it through this fully connected, two fully connected layers, so that the output of this branch is of length 256, right? So this is like the Euclidean subspace, and we are essentially embedding each spectrum into a Euclidean subspace of size 256, right? And then we do the same thing with the peptides. So if you have worked on deep learning before, you'll probably notice that this is a straightforward thing to process sequences, right? LSTMs are really good processing sequences. So since each peptide string here is a sequence of characters, right? We process each peptide using bidirectional LSTM. And then again, we get a feature vector here, and then we have kind of similar network of fully connected layers here. And at the end, each peptide gets embedded into a subspace of 256. Now what's happening here is that we are taking data from two different modalities, right? We have floating point arrays and we have character strings. And we are embedding them into same subspace, right? We're of similar length. And comparing floating point arrays that are of similar length is much easier than comparing a floating point array with the character string. So you can think of it as those algorithms that take an image and generate captions, right? Or take captions and generate images. Or a more suitable example would be an algorithm that takes an image and a caption and tells you whether this caption is suitable for this image, right? So you can think of this peptide string as the caption, and you can think of spectrum as an image, right? Image is a bunch of floating point array numbers, right? Or well, integers, right, of a 2D array of integers. So it's somewhat similar to image caption comparison. So yeah, this is just the information of how we actually do the processing. So we kind of translate this spectrum into an array of length 80,000, which is a bit much. So we're working on reducing the size of this array. And as I mentioned, this intensity or relative abundance is not that important. So we just normalize it um, into values between 0 and 1. And then we encode the peptide strings into unique integer assigned to each amino acid. So there are 20 amino acids, so you just assign a unique number to each amino acid, and each peptide string becomes an array of integers. And then you do an embedding layer um, and learn an embedding for each amino acid. So each peptide ends up becoming a 2D array of floating point numbers. And then that is fed to the bi-LSTM, and then it processes that 2D array. And there are a bunch of other things that we kind of augment to this network. I'll just skim over them. So there are auxiliary branches, which kind of nudge the training process of this deep learning architecture towards better learned parameters. So the way you do it, you make the network predict a bunch of relevant things to a spectrum. So it predicts the charge, mass, and this branch predicts the mass of a peptide. And then we do auxiliary, not the auxiliary, adversarial branches. So have you guys worked with generative adversarial networks? Or if you know about them, you have heard about it, right? So the way they work is that you have two networks. One is generative network, is generating some data, right? And then there is an adversarial network that is predicting whether the data that's coming in is actually a real data or generated data, right? So they kind of fight with each other. The purpose of adversary is to detect whether this data was generated or is a real world data. And the purpose of the generator network is to fool the adversarial network into misclassifying its own generated data set into real data set, right? And that's how you end up generating or data set that looks almost as good as real data set, right? So here what we do is, our goal is that we want to train these two branches so that the embeddings, these embeddings from both branches look similar, so that the comparison is more meaningful. So we use an adversarial branch, which is mainly detecting whether this embedding came from the SSN branch or the PSN branch. And the way the training happens is that you take a spectrum and the corresponding labeled peptide, and in the embedding subspace, they are closer to each other. And the way we measure the distance is L2 distance. So we have two arrays of length 256, right? And you measure the distance between them using L2 distance, right? And the goal is that if a peptide belongs to a spectrum, their distance in the embedded space should be lesser than the other peptides or spectra. So for example, here you have Q1 and you have P1. And you do pre-processing, and it goes to the SSN and PSN branch, and the learning processes embeds them closer to each other. So you end up with these pairs of length 256. So that's how we can measure the similarity. So what we do is we just pick the closest peptide to a spectrum, and that's highly likely to be the correct one. So this is the loss function equation. It's similar to triplet loss function, but I'll kind of go over it. If you are interested, you can approach me after the presentation. I'll be happy to kind of like give you the idea of what's happening here. And then, so these are the results that we got. The initial results, which were really promising. We got the ROC curves for the accuracy of the measurements, and this green one is our algorithm. So in ROC curves, if the curve is to the left and upper side of the graph, it's better performing algorithm. The other way to measure is the precision recall curve, where you kind of plot the precision against recall, and if the curve is to the right and upper side of the graph, the algorithm is better performing, and we compared it against the state-of-the-art X-score and hyperscore algorithms, and our ended up significantly overperforming than those ones. And this is another way of measuring the accuracy, when how many actual peptides you are able to identify in a real world scenario. And in this case, we were close to the traditional algorithms, but in some cases we did end up performing a lot better than X-score. So this is what we are currently working on, improving the database search accuracy. So yeah, that's the project that I'm working on. So I kind of like gave you the in-depth project. So if you have any questions regarding this project, I'll be more than happy to answer any questions. The next one is the same project, but the high performance computing version, or parallel computing, where we actually take the protein database and we distribute it into different subparts of the database, so that we can distribute these sub databases onto different nodes on the supercomputer, and the processing that happens is load balanced, and there is the communication is minimized. So 𝐒𝐮𝐩𝐞𝐫𝐜𝐸𝐴, who is working on this project, he designed a load balancer. He optimized the localized data processing, so the communication is minimum, and then he designed a dynamic resource managing algorithm, so that to measure the accuracy, latency, or to improve the latency of the algorithm. And we are using the 𝐒𝐮 supercomputers for this purpose. And these are just the results that we got. We have a pretty good strong scale efficiency of the algorithm. Strong scale efficiency is how good is the algorithm performing when you increase the number of cores, but the data type stays the same. And then we have the speed up, which is pretty close to an ideal speed up that you can expect with the increasing number of nodes. So this is the same process. This is the same algorithm for identifying peptides using spectra, but on a distributed system. And so that's pretty much the wrap up of all these three projects. Well, the two projects, the third one is coming. So why is this all useful? So recently CPTAC published 19.4 terabytes of data related to a mass spectrometer and the peptides. And this is a huge amount of data that needs to be processed, ideally in real time. But because the data set is huge and our computational abilities or resources are so limited, that a lot of this data ends up never properly looked at or properly processed. So it's called the dark matter of proteomics. It's just a data that's just sitting there. And this is a problem with a lot of other data sets, right? It's become a cold data. which is just sitting there on a server somewhere and probably ignored forever. So, so that's one of the reasons that high performance computing and machine learning techniques are becoming so popular, because we have so much labeled data that we can train bigger algorithms, but we need high performance computing so we can analyze or process all that data that's sitting there. And then is the third project, which is extremely energy efficient machine learning using reconfigurable computing. So that's FPGAs. So if you have interest in FPGAs or you have worked in FPGAs, this would be a really interesting project for you. So what we are doing is we are designing machine learning architectures, convolution networks, LSTMs, and other networks um, that are configurable on FPGAs. So the benefit of this method is that these algorithms would be highly energy efficient compared to the ones that run on GPU, and with a decent speed up. So yeah, so if you have interest or experience working with FPGAs, this would be a really interesting project for you. So that's all the details related to the projects that we are working on, and we are looking for PhD students in all these fields. Your experience that we kind of need is that we have strong algorithm design skills, background in machine learning, high performance computing is preferred, but it's not necessary. The same goes for background in bioinformatics. If you have some, it's good, but if you don't, then that's okay. So I didn't have any background in proteomics or any of these, so I spent some time doing literature review of these methods, of the algorithms that are already developed in proteomics, and then I ended up kind of learning more or less what was needed. And then doing PhD at FIU, and what are the advantages and why you should uh, join PhD. So these are the slides provided by the department to us. First of all, PhD in US is not an academic only kind of outlook. A lot of students end up working with industry because so many companies are working on research in US, and you end up getting paid a lot. So just to put it out there, you will get a lot of money if you finish your PhD in US and end up working for either a corporate or in a research lab, and the median salary is 122,000 per year, which is pretty decent compared to what you get for undergrad or master students. And this is the graph that highlights that the 5 years that you will spend doing PhD will put you ahead of undergrad student who has been working for 5 years. Because so this is the salary increment for an undergrad student, right? And you can see that that student is going to get there in 9 years, where a PhD student will start, right? So you will be 4 years ahead of an undergrad student who's been working all the time, the whole time. And this is not just figures. One of my colleagues, he was working in a different lab, but he was working in bioinformatics. But he ended up working on machine learning projects, not related to bioinformatics, just because he had experience in machine learning and deep learning, and he's getting paid 160,000, and he's working with Facebook. So, yeah, talking in terms of money, you will be way ahead of undergrad or master students. So this is kind of like one of the concerns that the PhD students have, that they don't get paid enough, but which is no longer the case. And more PhD students are needed in the industry now. And yeah, so the other things you're probably already working on research. So you will be ahead of the curve in terms of latest technology. So every time there is a new technology in deep learning, machine learning, high performance computing, you are aware of it, right? So there is an issue of kind of lagging behind the industry when you're working on your PhD. You will be at the forefront of the research. Then you can, there are opportunities, there are startup opportunities within FIU. So that's another good thing. My advisor has talked about doing a startup, but it's up to the person. If you want to do it, there's also an opportunity. And then there are a lot of research labs. One of my colleagues is working in Berkeley National Lab. Another one did an internship recently there. Or eventually you can end up working in say software engineer, but usually you end up working as a scientist in industry. So if you are interested and you do end up applying, like what are the things that you can do? You can contact me, Dr. Said. I'll give you my email address in a later slide. Do go to our website and read research papers. You can reach out to me, Dr. Said, or any person in our lab. So I'll give you that information shortly. And yeah, talking about research at SCIS, that is School of Computing and Information Sciences at FIU. It has been growing a lot for some time, and I've seen that while working there for 2.5 years. We have a lot of PhD students there, probably more than 100 graduate degree seeking students. I think this is an outdated information, but yeah, we have a lot more than that. And we have so many, we have actually 3 professors who are working on bioinformatics there. And National Science Foundation, NSF, ranked us 43rd in research expenditures. And there is another ranking of provided by Carnegie Mellon, which ranks universities in R1 and R2 ranking. So R1 ranking universities, there are I think only 100 in US, are the ones that are focused on research based on their expenditures and the research quality that they produce. So FIU has been an R1 university for some time now. And then, the professors are good. There are a lot of labs. All of these labs we have there. Ours is kind of like bioinformatics, high performance computing, parallel computing, machine learning. So we're working on all of these things. So one of the good things about applying for PhD right now is that GRE is not required right now because of the pandemic. So which was a huge hurdle when I applied for PhD, that I had to take GRE. I had to prepare for it for months. So this is a pretty good deal right now that you can get admitted without a GRE. These are the GPA requirements. Honestly, these don't really matter much because we have students who have GPA 2.7 and we have students with 3.9 GPA. So Dr. Said doesn't really care about the GPA as long as you can do good in the interview. So if you have good CV and you can do good in the interview, then the question of GPA is not going to be there. If Dr. Said accepts you in our lab, then the department is not going to object on any of your GPA or anything. The only thing that you will need is TOEFL or IELTS, and these are the score requirements. So it's 80 for TOEFL, I think, and 6.5 on IELTS. And I don't think we have paper-based TOEFL here, right? We usually do online. I did online, so I think this is a more relevant figure. And if you are interested, these are the details. You can send your CV to me personally. I can kind of look at your CV and do a recommendation. but you will also send your CV to Dr. Said. This is the email address of Dr. Fahad Said. This is me. So if you are interested in working on projects related to deep learning, machine learning, proteomics, you can send me an email, ask me any questions, send me your CV. I can review it. I can tell you things that will help you in your interview, in your application. I can guide you through all the process. If you are interested in distributed computing, parallel programming in proteomics, my colleague Muhammad Hasib is working on that project, so you can contact him as well. So yeah, we'll be more than happy to help you out. Dr. Said told me that get him at least 2 PhD students and 1 postdoc student, but I think he will end up hiring probably more than that. In terms of money, we are funded lab, so we got $500,000 from NSF a couple of years back. Few months ago, we got $1 million funding from National Institute of Health. And this funding is going to be available for 3 years. So there isn't any problem, because when applying for PhD, it's always a question whether your professor is going to fund you or is he going to leave you in the middle. So, and usually once you get funded with the decent funding, right? So usually it continues. You end up getting more fundings. So money in terms of funding is not an issue. And this is our lab. We had a bunch of other students from Iran and other countries, but they recently graduated. So these are the people who are currently working in our lab. This is a web page of Dr. Fahad Said, if you want to note it down. And this is our lab's web page. And you can follow Dr. Said on Twitter to see what are the current kind of things or projects that we are working on right now. So yeah, that's pretty much the presentation. If you have any questions, you can ask me right now, and I'll leave it at this slides if you want to kind of note down the information. So yeah, thank you. इसका क्या? ऊपर करो, hold on. Yeah, the volume is full. Yeah. Is the speaker off on my end?
So, Usman, can you hear me?
You need to turn on the speaker? I don't know if it's not working. Okay.
Hello, Usman, can you hear me?
I think I cannot hear anybody if they're asking a question. Like the speaker is not turning on. Can you say something? So
I have a question if you can hear me.
ये माइक स्पीकर चेक कर लिया, वहाँ से स्पीकर तो नहीं बंद किया? अच्छा, स्पीकर तो ऑन है 100%. I guess they can type the question if they want in the chat. Okay, it's working now?
जी आवाज आ रही है आपको?
It's working now? Okay. Okay. So it's working? Okay.
So Usman, can you hear me?
So disconnected and speaker. Sorry.
बिल्ट इन पोर्ट इश्यूज कर रहे हैं हेडफोन नहीं वर्किंग कर रहे स्पीकर भी ठीक है आपको आ रही है
Actually, if anybody has questions, they can just type it here. I can answer them because they can hear me. Ataur Rahman, if you're talking about machine learning for PhD, then you can learn that during your PhD as coursework because you have to complete 30 credit hours um, in first 2 years of your PhD. And there are a lot of machine learning and AI courses, so you shouldn't be worried about it as long as you have background. You have background in biomedical engineering, but also more preferred um, skill is algorithms. For masters, We're not offering, I think, any master's positions in our lab. But if you just want to learn machine learning, I think there are a lot of online courses that I've seen on Udacity and other websites. Yeah, but if you want to do masters at FIU, it is very difficult to get funding for just masters. Screen share?
Yes.
If you guys have questions, you can ask meanwhile.
Is there any deadline?
For applying, I would say apply as soon as you can, because the application process starts in December. So you're all, well, I am already late, but it shouldn't be a problem as long as you get accepted in the lab. Because on Dr. Say's recommendation, they can still accept your application and process everything quickly, and you will still have time to apply for visa and get everything sorted. It'll be fast, but that's how I did it. I got my final acceptance letter, I think in June, and then I applied for visa, and I think I got my visa on August 7th, and then my flight was on August 10th. So yeah, it's going to be a pretty hectic, but it's not an issue. But to be in the safe zone, I would recommend that send an email to Dr. Said or me with your CV as soon as you can, because that's the first step, and then you can go further. And also have your TOEFL or IELTS completed.
Primary application, we have to with TOEFL or IELTS.
With what application?
With primary application, the initial application.
Yeah, you will need TOEFL score when you're applying online for the department. For sending your CV to Dr. Said, you won't need a TOEFL score or anything. It's better if you have it. You can mention that in the email, but it's not needed. But you will need to put in your score, official score, in the online application. So that's why I'm saying that have it kind of sorted out as soon as you can.
I will go for TOEFL or IELTS after interview with Dr.
You can do that, yeah. Big biomedical data. Yeah, so big biomedical data is it's different fields. Depends on what are you talking about. So if you're talking about genomics, there is the next gen sequencing where you get short DNA reads in terabytes. If you're talking about proteomics, you get terabytes and terabytes of mass spectrometry data. The word big generally refers to algorithm design when you have to consider the size of the data as part of your design strategy. Definitely doesn't fit all of it in memory at one time. And the amount of data that has that's being generated in these two fields, and also in connectomics, the fMRI scans, is reaching to that point, so that the algorithms that we design, almost all the tools or software that we use, consider the size of data and do out of core programming. So that's what I mean, that it's a big biomedical data. Hope that helps. database here, all data regarding. Which work are you talking about? You're talking about the biomedical data. There are multiple online repositories where you can kind of store your data and retrieve it whenever you want to. Okay, so if you have any more questions, I think you can send me an email. I'll be more than happy to assist you or guide you through the process or answer any of your questions. Or you are more than welcome to send your CV to Dr. Said himself. So I'll leave this slide here. So these are the email addresses. So yeah, I think we can wrap up, right? All right. Thanks everyone for attending the presentation. Yeah, looking forward to hearing from you guys.
Thank you, Mr. Usman. And I think we should wrap up this. If you have any questions, you can contact directly to Mr. Usman and other email addresses that are given to you. Thanks for joining.